My name is Andrew Needles from Visual Sonics. It's a pleasure to introduce my colleagues today, Mr. Dieter Fuchs from our office in Amsterdam. Dieter's our Market Development and Preclinical Applications Manager. Dieter joins us remotely today online, and Dieter, it's a pleasure to have you. I'm also joined by our Product Manager, Andrew Heinmiller, Product Manager for Photoacoustics. Drew is here with us in Toronto, and Drew, it's a pleasure to have you joining us today. So again, my name is Andrew Needles. I am the moderator for today's session. At this point, though, I will be handing things over to Drew to take us through the agenda for today's session. So thank you, Drew.
Great. Thanks very much, Andrew. Hi, everybody. Thanks for joining. I'm just going to really quickly go over what we're going to be speaking about today before launching into the introduction first to multimodality imaging. I'll give a very brief introduction there to the different modalities and sort of strengths and weaknesses of those modalities. Then I'm going to introduce our Vivo high-frequency ultrasound and photoacoustics products, describe how they fit into the multimodal imaging as far as the pros and cons. Then I'm going to hand it over to Dieter, and he's going to talk about multimodal imaging with the Vivo systems with specific application areas in mind, namely anatomy and volume, blood flow and perfusion, ischemia and hypoxia, and molecular imaging. I'm then going to take the mic back and talk specifically about our products and about how they fit in and how they can be used for molecular imaging with some photoacoustic contrast agents. I'm going to end off talking a little bit about data management with the in vitro product. So first, just again, a very quick introduction to multimodal imaging. There are certain requirements for these in vivo imaging requirements, what we've called them. One is fairly obviously reliability and reproducibility. This is obviously very important when using any kind of imaging modality, in vivo or otherwise. And then there's validation. There have been some modalities that have been around for a long time and have been validated against different modalities in vivo and otherwise. And Dieter's going to talk a little bit more about that. Then there's the sort of common advantages, things like speed, ease of use, clinical relevance, and also being multi-purpose, being able to look at a wide variety of applications as well. So those are the basic requirements. Now I'm going to talk a little bit about specific modalities here. So I'm going to talk about PET-CT, MRI, and optical. I just want to point out that this is not a completely exhaustive list of pros in the green and cons in the red, but just a very general overview. And I may miss a few things, and please feel free to add or comment on this in the chat window on the right-hand side there. First of all, with PET-CT. With PET-CT, and I'm not going to describe the principles behind these different modalities, just where they fit in terms of their advantages and disadvantages. For PET-CT, there is functional and anatomical information available. It's great for doing whole body imaging, and you're able to image through the entire body of the animal, or indeed in the clinic of the patient. There are limitations because of cost. These are expensive modalities typically. They require a certain amount of expertise, especially when you're handling some of these PET reagents. It's very specialized procedures for doing some of these experiments, and of course it does use ionizing radiation, so there are limitations as to the doses that your animals are able to handle here. Moving on to MRI, you have great soft tissue contrast with MRI. It doesn't use ionizing radiation, and you're able to obtain functional information such as blood oxygen level dependent MRI, functional MRI. Cost is also an issue here. Some of these these procedures and scans in particular can be time consuming, and therefore a lot of these are not necessarily real time. It's not really a real-time imaging modality. Moving on to optical. Optical is extremely sensitive. These cameras can detect single photons. So it's very, very sensitive modality. It's quite easy to use. I won't mention the product names here, but you can picture these here. You open up the door, you put your animal in, and you set the camera and start taking photos. So they're quite easy to use. And for optical, there's many different probes available, fluorescent probes, to be able to tag different things and do molecular imaging. The limitations with optical, the resolution at depth is quite poor because of the scattering of light in tissue. That also makes it very difficult to accurately quantify especially signal that's deep within the tissue. It can also be limited to specific cells if you're doing things like reporter imaging. I'm going to talk about in the same context, ultrasound and photoacoustics. Ultrasound has decent soft tissue contrast, again, doesn't use ionizing radiation, and it is truly a real-time modality. You can place the probe down on a patient, or in this case, when we're talking about research, an animal, and you can see the live beating heart of a mouse, for example. It's not optimal for some tissues, such as bone, so the ultrasound doesn't penetrate the bone, so there are some limitations there when compared especially to something like CT. There's also some training required, although this modality is meant to be used by anybody. Especially in the research realm, anybody with a little bit of training can get up to speed and can use ultrasound. As opposed to something like MRI or PET, where it really does take an operator, a dedicated operator, who's really an expert at those modalities. Photoacoustics actually shares the same advantages and disadvantages with ultrasound, but in addition, as far as the advantages go, you get the resolution of optical signals at depth. So we can actually resolve those signals because we're detecting the ultrasound that's coming back, even though it's originally from an optical absorber that we're seeing. We do get functional and molecular data as well, and I'm not going to go into that. I'll let Dieter cover some of that. The limitations, there are limitations in sensitivity if you're comparing it to optical. You're not just looking at single photons coming out. You're looking at the conversion of light into heat, and I'll describe that a bit later, and then detecting the ultrasound wave that comes back. And then there are also limitations in depth compared to things like MRI and PET-CT, which you can go through an entire human being, where there are depth limitations with photoacoustics, where we can see through an animal, but when you start getting deep into patients, things are a little different. I'm going to talk about the Vivo products now. First of all, the high-frequency ultrasound. And for those of you who are not familiar with us, we do indeed make high-frequency ultrasound products. So in the clinic, you'll typically see frequencies between 3 and 15 megahertz. We're using frequencies 30 to 80 megahertz. So on the right-hand side of that slide, you can see there's a human fetus on the top one using a clinical system, and then on the bottom image, it's actually a mouse fetus. So this is a tiny thing, and you can see that the resolution is approximately equal in those two images. So with high frequency, we get this very, very high resolution, which makes it ideal for doing in vivo small animal imaging. For photoacoustics, I want to talk about the photoacoustic effect. First of all, photoacoustics is really a hybrid of optical and ultrasound, where we use a pulsed laser light. So we use this optical signal, we illuminate a tissue. Anything that absorbs that light will heat up a little bit, give what's called a thermoelastic expansion, which creates a pressure or a sound wave which travels through the tissue, and then we detect that with ultrasound to create the image. That's the long explanation. The short explanation is light in, absorption of that light, and then sound out to make that signal. And we'll go through this again as we go through the presentation. These are our sort of products here, our main products. The Vivo 3100 is the brand new ultrasound platform. It's got a touch screen. It's a significant change from the Vivo 2100, which you can see actually on the right-hand side. But the Vivo laser, the platform, is the 2100, where we've got the laser attached to be able to do photoacoustic imaging. And again, I want to sort of point out some of the specific advantages of our systems in particular, not just the ultrasound modality, but these systems are, as I mentioned, high resolution. So we're able to see, orthotopic tumors, for example, micro-distribution of different contrast agents. So that's the really, really strong advantage here, is being able to get that resolution, which you may not be able to get with other modalities. Also, it's a fairly open platform allowing interventional procedures. We've got an ultrasound, sort of essentially a handheld ultrasound probe, which you can see actually hanging off the 3100 on the left side there. So it allows some interventional procedures. It's also quite high throughput. You can image many, many animals in a short time. And really, we're talking about this is a multi-modality webinar where we're talking about other modalities, but even within these platforms, there are multiple modalities there, and I'm going to let Dieter expound on that. There's also the 3D rendering capability as well, and I think Dieter's going to demonstrate that. At this point, I am going to hand it over to Dieter, and he's going to talk about specific applications within both the Vivo systems and other modalities.
Thank you, Andy, for this overview of different imaging modalities. I'm now covering a little bit more the essence of multimodal imaging. Let's look at this image here of an iceberg. I like this analogy of looking at an iceberg because let's think about tumors. You look at tumor volume as it changes. It's a single parameter. It's very important, but in the big picture of the complex disease of cancer, it feels like you're looking at the tip of the iceberg when there's so much more to it than a volume. Of course, as I mentioned, it's essential, but you could also look at perfusion as in terms of response to therapy, looking at ischemia, hypoxia, and a little bit more molecular imaging on the tumor. We're going to cover all of these points in the subsequent slides. First of all, let's stick to the example of tumor biology. It's important to detect tumors, or in basic principle to look at anatomy. And that's to do that in 2 dimensions or also in 3 dimensions. So we have here two movies. I hope you can see them play. So on the left-hand side, you have an ultrasound image of a 2D scan. We're looking at the left side of a mouse abdomen, and based on the tissue density, you get the sound waves coming back, and you see it outlined. For example, here a left kidney, labeled in yellow. Right on top of it, you see a spleen. You have a little bit of pancreas tissue in there. Just in the 2-dimension section, together on the ultrasound, you can link this with a 3D motor to do a 3D scan across the abdomen, as you can see now in this movie, and you get information on tumor volumes, for example. What hasn't been mentioned so far is that physiologic monitoring animal physiology is important in imaging if you look at functional parameters. And at the bottom of this 3D scan, you do see respiration and ECG displayed, and also a gating for respiration for doing a 3D scan on the ultrasound. What has been done to looking at such a cross-section in the mouse is, for example, for detecting pancreatic cancers. And on the next slide, you can see a publication from 2013 looking at tumor and pancreatic tumors. And let's see, moving to the next slide, yeah. Where the optical imaging was used for quick screening purposes, excellent tool for that. And also MRI for looking at tumor evasion into the retroperitoneal space, but also the ultrasound for detecting the smallest and tiniest of tumors, and the tiniest tumor in that study that was detected had a diameter of below 900 micrometers. On this image on the bottom right, you can also see red structures. These are larger blood vessels, because with the ultrasound detecting the echo, you see the anatomical structures based on density, but looking at a shift in those sound waves, you can also detect the motion. And suddenly you end up with information about tumor location, possible infiltration, tumor size in terms of volume, and also whether this tumor has larger blood vessels inside it. All of this very relevant information. If you're interested to look more into pancreatic tumors, I can recommend this publication, this book chapter from Sastra and Olive. What is very important for this here is also that if you have models for spontaneous tumors, or you lose patient-derived tissue samples that you implant into your animals, you can still detect them with ultrasound, because what you detect is tissue density. You don't need any kind of secondary reporter, enzymatic activity, etc. No additional contrast needs to be injected. Andrew mentioned initially that you need validation of your findings, and there are very good publications looking at ultrasound, MRI, optical imaging for all they can do and all the limitations they have. As for the ultrasound, detecting pre-palpable tumors is one of the key benefits, as we had before in the publication on the pancreatic cancer. 3D visualization and quantification is also a key feature of the ultrasound. And with this publication from 2011, Theis et al. Looked at three-dimensional images from the ultrasound and correlated this data with images derived from an MRI. And it's very clear to see that you have a very nice linear correlation. And on top of that linear correlation, if you have 1 cubic millimeters on an ultrasound image, you have the same on the MRI. So a nice validation for either imaging modality. In addition to that, whatever you do, whatever you measure should be reproducible. And the publication from 2010, you can see that the mean deviation of multiple 3D measurements in that case is below 2%, so very high reproducibility on the ultrasound. In another study of an orthotopic tumor model, again in a pancreas, this publication compares what you can do with the ultrasound and optical imaging, with all the benefits and drawbacks. So for the ultrasound and optical imaging, both were able to detect the tumors, but what has been done in addition to a surgical implementation of tumor cells into the pancreas was that under ultrasound guidance, some mice received the tumor cells without surgery. And here the ultrasound could prove that you can use it both for detection of tumors, as before, but also for actually implementing or injecting tumors into the animal to avoid surgery, as the surgical procedure itself gave a false positive signal on the optical imaging. With image-guided injection, I'd like to show that on the next slide, where you have now highlighted on the right side on the screen. You see a tumor highlighted in a pinkish color, and inside the tumor you have some fluid and debris floating around. And under ultrasound guidance, you can go into that tumor and either inject your therapeutic agent, as briefly shown here, or you make an aspiration, take fluid samples, take biopsies with minimal invasiveness. And you can do this in addition to looking at the tumor volume, infiltration, spread, etc. This is so far for pure anatomical structures, but there's more to that in a tumor or other organs, namely the blood, the perfusion, the flow. On the ultrasound, you can nicely see this by using contrast agents. If you want to look into the capillary flow, contrast agent at the size of erythrocytes, meaning the contrast agent does stay vascular, does not diffuse into tissues. You don't need to correct for diffusion parameters. And you can also use this contrast agent for molecular imaging. So the insert on the right is not drawn to scale, but you can use antibodies to label your bubbles or contrast agent for detecting endothelial surface markers. Moving to looking at perfusion and flow, this is a model for a brain tumor where the ultrasound is used for non-invasively assessing the flow using a Doppler, the center bottom of the screen, or using contrast agent to looking at the perfusion and the capillary function inside the brain, both healthy brain structures and the tumor. What Andrew mentioned before on photoacoustics, you can also use the combination of optical and ultrasound imaging to, in that case, look at tumor oxygenation or oxygenation in the entire brain. So no craniotomy, etc., had to be done for getting these images. Excuse me. And you can thereby assess hemoglobin, whether it has bound oxygen or hasn't bound oxygen. To go a little bit more into detail on this, I'd like to go for the hypoxia ischemia session. So again, all the images I'm going to show now for tissue oxygenation, etc., comes in addition to a tumor volume, infiltration, metastasis, blood flow, capillary function. So getting more of the whole picture, seeing the entire iceberg. For looking at the oxygenation levels, the tunable laser on the photoacoustic system allows you to look at multiple wavelengths. And for the oxygenation, we decided to go for 750 or 850 nanometers, apply some math to this, and you get parametric images showing you oxygenation saturation inside the tissue. In this study, there was a comparison looking at photoacoustic imaging in a tumor and compare that to bold MRI, so the blood oxygenation level dependent MRI images. And both showed a very nice correlation to each other, and you didn't need any kind of contrast agent for looking at the photoacoustic signal inside the tumor, and you got nice parametric images for both imaging devices for whether the animal was breathing air or carbogen and going back to air. The main difference between the bold MRI and the photoacoustic is that photoacoustic imaging you could do longitudinally continuously throughout the study, whereas the bold MRI you do at different time points. This continuous imaging allows you to do dynamic, to assess dynamics inside a certain experiment. Brings me to the next publication in stroke, looking at a model for brain ischemia induced by tying off a vessel, forming a thrombus, and then looking at oxygenation levels and flow inside the brain. So you see on the top left, you see an image of with a laser Doppler showing you the superficial structures of the brain with stroke, top saline, and the bottom image is with the drug removing a clot, so see you get reoxygenation levels in the tissue. To go deeper, however, the laser Doppler is not enough. You would need photoacoustics. And as for the experimental setup, you can see that the animal is going on an invasive procedure. Common carotid artery is tied off, stopping the blood flow specifically in one hemisphere on the brain. After some time, it is released to allow a reperfusion in the tissue. Now, when looking at this, after tying off the common carotid artery, you see that one hemisphere remains highly perfused as indicated by a Doppler signal in red, whereas the other half of the brain, larger blood vessels no longer have blood flow inside. Whereas looking at with photoacoustics at the hemoglobin oxygenation levels, you see that both hemispheres still have oxygen levels, just that the right side has lower levels. And you get this information as opposed to the laser Doppler also at depth throughout the entire brain. Now, this contrast agent, hemoglobin, it's an endogenous contrast agent, but of course you can use the photoacoustics with other contrast agents, some exogenous contrast agents. There are a number of those available, and Drew will go more into detail in that. I chose some slides on ICG, clinically approved agent for looking at inflammation or infection. Bacterial infection after surgery are a clinically relevant problem. And what has been done in this mouse model is that ICG-labeled bacteria were injected subdermally. And if I use my pointer, let's see, no, it doesn't. So my pointer, you see an injection site here for the bacteria, but you also see a photoacoustic signal for the ICG throughout, in that case, the hind limb of the mouse. This imaging was also done in addition to photoacoustics with bioluminescent fluorescence, where you get a bright signal from the injection site, but you don't see what's going on in detail in the leg. Whereas on the photoacoustics, since you co-register your signal from the ICG with the anatomy, you do see the injection site, but you're also able to detect the lymph node and to see bacterial spread from the injection into the lymph node and posterior part of the leg. Different contrast agents one could use also reporter genes. And this specific study uses tyrosinase as a reporter gene for multimodality imaging. So tyrosinase producing melanin used to be detected with MRI, photoacoustic imaging, and also with PET, positron emission tomography. Very interesting study, as the melanin has a very broad photoacoustic spectrum, so you can specifically detect it and differentiate the signal from the blood. The MRI uses the ability of the melanin to carry iron, and for PET, a different melanin-specific contrast agent was injected to be able to detect it. Again, the photoacoustic allowed you to image expression of tyrosinase over time in a continuous study in case you have a condition knock-in, knock-out. PET required you to do a longer imaging session. It's indicated here by the 2 hours time span from time of injection to allow clearance and see the accumulation by detecting the tyrosinase. With that, a very brief introduction into different imaging modalities, different things you can do with ultrasound and photoacoustics, and I'd like to hand over to Andrew, to Drew, to give a little bit more information about various photoacoustic contrast agents.
Thanks very much, Dieter. Dieter's, talking about doing some of this molecular imaging kind of gives me a nice segue to talk about contrast agents. For example, in the bacterial infection study where this antibiotic was attached to this optical dye, ICG, to label infection. And also the reporter gene where you're using melanin as a contrast agent. I just want to go into a little bit of detail. These are both, we can see both of these with our photoacoustic imaging system. So I want to go into a bit of detail about some of the contrast agents, and I want to start with an example of a multimodal imaging agent. So this was in Nature Nanotech, a paper in Nature Nanotech from Gang Zheng's group, where they were looking at a microbubble. So Dieter also described these microbubbles, which we can use for ultrasound contrast to see, perfusion right down to the capillary level. These are basically a lipid shell with a inert gas inside. So what this group did is they took a microbubble and they were able to actually convert that microbubble into a nanoparticle. That microbubble had molecules in it that absorb light as well, and when they become nano-sized, they actually absorb more light, which means, if you remember at the beginning of the presentation, the principle of photoacoustics is really optical absorption within a given wavelength range. So I just want to bring your attention here, and let's see if I can find this. I liked Dieter's pointer there, so let's see if I can find a laser pointer. No, that's not it. Oops. Here we go. All right. So I want to bring your attention to this figure here. I mean, this is a principle that they're using where you have a relatively large microbubble, probably 2 to 3 micron, around that size range, and you're converting it into something 5 to 500 nanometers, so truly a nanoparticle. And I want to bring your attention here to this figure here. It's hard to read, small text here, but you can see this is the ultrasound signal. So when you've just got the regular microbubble here, we get very, very nice acoustic contrast. So just using nonlinear contrast mode on the system, you can see this very, very nice acoustic contrast. When you convert these bubbles, you actually lose the acoustic contrast. So these are pulses of this ultrasound that they're using to convert these microbubbles. And as you convert the microbubbles, the signal from the ultrasound disappears. The next panel down is the photoacoustic signal using our Vivo laser system, where, in fact, as you convert these particles into nanoparticles, these microbubbles into nanoparticles, you actually get an enhancement of the photoacoustic signal. And then, of course, the molecule that they're using within these bubbles to absorb light is also a fluorescent molecule. So you can see the fluorescent signal actually increases a little bit as you convert these things. But now you can see here that we've got a true triple modality agent here, where you've got ultrasound, photoacoustics, and fluorescence. The implications of that are many, one being this could be a potential drug delivery mechanism, and you could actually image how much of that drug is being delivered based on maybe the decay of the ultrasound signal or the increase in the photoacoustic signal as you convert these things to nanoparticles, where they're small enough, basically, that they'll be able to escape the vasculature. Again, I'm not going to go too much into the implications of all this, except to point out that really we've got 3 modalities now, and this gives us flexibility across not just our own Vivo platform, but across other imaging modalities as well. And I want to focus specifically on the photoacoustic contrast agents, not just because I'm the product manager, but this imaging modality lends itself well to cross-modality agents, as I just described. So things like nanoparticles. Nanoparticles are used also in MRI contrast studies as well. We can use them with photoacoustics as well for things like cancer imaging and therapy, really doing molecular imaging within the cancer imaging and other application areas. NIR dyes is a pretty obvious one. So near-infrared absorbing dyes exist already for doing fluorescent imaging, namely something like IR800CW made by Lycor, which is actually currently in clinical trials. These things there's a great potential for these things. They're small, they can be eliminated by the body, and incidentally, they can also give a very nice photoacoustic signal. So we can do things like, in this example, you see here this green signal, for doing cell tracking. Those are labeled cells that have been labeled with a near-infrared dye and injected into the hind limb of an animal. And I'll describe some of an image like this a little later on. Won't go into too much detail about it here. Liposomes are another one. So the paper I just presented was using a liposome-based system for doing things like drug delivery, for example, and then Dieter talked already about using melanin as a reporter for doing genetic reporter imaging. I want to talk about some of our partners in some of the contrast imaging. First, there's a company called Optimios who has licensed a technology from Princeton for a polymer dye-based nanoparticle. Again, now we've got this polymer, it's got a dye in it, so we're able to see it with photoacoustics and potentially also with fluorescence. So it can be a multimodal agent, and now we can functionalize it with small molecules and so on to do true molecular imaging. We can also have multiple agents targeted to different targets because you can customize these so that they basically have, they look different. They're different colors and they absorb at different wavelength ranges, and we can use our Vivo laser system to separate them. And again, I'll talk about that in a couple slides. Nanohybrids is a company based out of Austin, Texas, who makes these gold nanoparticles, specifically silica-coated gold nanoparticles, give an extremely high signal in photoacoustics, but they, again, can also be used with other modalities such as MRI. And these, again, can be functionalized for doing that kind of molecular imaging. Finally, Genelux has constructs to be able to do reporter gene imaging with using melanin as a reporter. This doesn't rely on contrast agent delivery, so that's one advantage of using this genetic reporter. It doesn't rely on delivering a contrast agent to a specific target or getting it there at all. It's just the sort of expression of the melanin at the target site. It's inducible as well. So if anyone out there is interested in exploring any of these technologies, please get in contact with us. We can give you more information. I want to summarize how the Vivo laser system can actually be used to do molecular imaging in this case. And it looks like this, and this may also look somewhat familiar in other studies, perhaps PET or MRI and optical as well. The first step here is to characterize a contrast agent, and this is the basic workflow for doing it. We have a sample that we can put it into a phantom. We then basically through one click we actually characterize what the sample looks like, basically what the absorption spectrum looks like of that sample. Then when we go in vivo, we prepare the animal, we select the components we want to scan for, we want to look for, and then we can scan. We can actually perform an injection. We've got an injector pump which is hooked up to the Vivo software. So we can, as we're imaging in real time, observe the uptake of a given contrast agent. And then of course we can scan in 3D as well to be able to get volumes. As far as the quantification goes, once we've acquired our 3D volumes at multiple time points, or even our live injection, we can then unmix the signals from the different components and quantify them as well. And I'll describe that in a slightly more detail here. So this is the characterization phase. So we're actually working on a few hardware improvements to be able to do this in a very high throughput way, where we're building a phantom where you're able to introduce different contrast agents, and again, as I mentioned, characterize them, find the peak absorption wavelength, the signal strength relative to other contrast agents, and also draw these sort of spectral profiles where you've got wavelength on the x-axis and the signal, photoacoustic signal intensity on the y-axis there. Again, going in vivo, we really have tried to optimize the workflow, so it's really just one touch. You identify what components you're looking for example, oxygenated, deoxygenated blood, maybe melanin, and then your contrast agent, your dye, nanoparticle, or liposome in there as well. And again, being able to hook up the pump so we can do the imaging at the same time as we do the injection for sort of perfusion parameters. And there are tools also in there for doing graphing and multiplexing and so on. This is one example of what this tool might look like, where you've got the nanoparticles, this is actually a study that we did in collaboration with Optimios, where we had just a sub-Q tumor model, but keep in mind that ultrasound is ideal for doing this kind of thing for orthotopic tumors. But this was really a proof of concept experiment where we wanted to inject two different kinds of nanoparticles, essentially, the green one and the yellow one. We inject these two different kinds of particles. One is targeted, one is non-targeted in this case, and then we can see the distribution of the targeted and non-targeted, but we also have other absorbers in there, keep in mind, oxygenated hemoglobin, deoxygenated hemoglobin, and melanin, and we're able to separate all these components and then display them multiplexed, like the image you see here with blue being deoxygenated hemoglobin, red being oxygenated hemoglobin, green being the non-targeted particle, and yellow being the targeted particle. So this is a really, really nice example of being able to look at multiple parameters. And again, of course, you've got the ultrasound in the background, so you're able to identify the anatomy and the microdistribution within the anatomy of these different absorbers. Finally, you've collected all this data. You want to be able to sort of compile all this data, report on this data, analyze this data, and potentially even compare it to the data you've acquired with the imaging modalities that you may already be using, such as PET, MRI, or optical. A way of doing this is using InVicro's software, their IPAX software. So we've got a partnership with InVicro where they've actually designed a custom package for their software. IPAX is essentially a web-based data management software for organizing, processing, and reporting on your imaging data. And as I mentioned, there's a package within IPAX now specifically for visual sonics ultrasound data. This allows for doing customized reporting, collating measurement data, and again, not just from the Vivo software and the measurements that you've done there, but also collating it with other imaging modalities. So, for example, if you've had an animal that you've imaged with multiple modalities, you can pull up the data from that animal, pull it together to do the analysis using the IPAX software. Again, if you've got questions about this, I please invite you to get in contact with us, and we can explain this a little more. That's all we've got for today. I think we're going to open it up now for questions.
Great. Well, thank you both to Drew and Dieter. Lots of different interesting applications that we've seen. Hopefully it's given everyone a sense of how the Vivo technology, the ultrasound and photoacoustics, can be used for a variety of different areas, including molecular imaging, which we really focused on towards the end. And in addition, its complementary aspects with other imaging modalities that many of you may already be using. So I would like to open things up for questions. The first one comes specifically related, a specific question about ultrasound and MRI, and are there any multimodal imaging agents available for these two modalities yet? Drew, do you want to maybe start off with that one?
Sure. There's lots of research going on, obviously, with design of these contrast agents, and how they can be used in different modalities. What I'll say is that, as far as commercially available agents go, there's obviously agents available such as microbubbles, which are actually used in the clinic for doing ultrasound imaging, and then different kinds of MRI agents. And the research right now is kind of focusing on designing ones that, as I mentioned, can image across platforms. I think as far as ones being commercially available, there are some developments, but a lot of this is still in the research phase, designing these things and optimizing them in the research phase. But we're sort of working with our partners to be able to see, how these things may be commercialized in the future.
Great. Okay. Thanks, Drew. Just while we're getting some other questions coming, and we'll get those organized, Dieter, a question actually for you, just about, we've seen a lot of preclinical imaging on the webinar today. Obviously, many of these imaging technologies are used clinically. Just wondered if you could say a few words about potential for clinical translation of some of the ultrasound and photoacoustics specifically. Dieter will just give you the audio controls here back.
Okay.
There you go.
Okay. Yeah, certainly. So ultrasound and photoacoustics do have great clinical potential. This is our just most important right up front. With these high frequencies, you really get very high, crisp resolution on superficial structures. Drawback obviously being penetration, but for superficial structures, you get real-time images at a very high resolution. Again, non-invasive, hand-transducer, etc. Same for photoacoustics. So optical imaging itself is not widely used, as you all know in the clinic. But with the help of photoacoustics, you get light into tissue and get an echo back. So you can then use this to visualize endogenous contrast as we had before, for example, the blood, or clinically approved contrast agents such as methylene blue, which works fantastically for photoacoustics, or ICG, as shown before. So there definitely is great potential in that direction for both modalities.
Okay, great. Thanks, Dieter. We've got another question. Perhaps both of you could give your ideas on this. A question about cartilage imaging. Can photoacoustics be used to image cartilage and other connective tissues in detail? So, Drew, why don't you start off?
Sure. I've actually done some of this imaging in basically just a demo situation where someone asked, can we see cartilage? We did imaging of the xiphoid cartilage, as well as doing quite a bit of imaging on the knee joint of mice. So being able to look at vasculature within the knee joint, and this is for osteoarthritis applications, also looking at things like lymphatic drainage, using ultrasound and photoacoustics to do that. And there was one particular example where we were doing image-guided injections of photoacoustic contrast agents, a dye, indocyanine green, that ICG dye that was mentioned in the bacterial imaging portion. Just injecting ICG, image-guided, into the synovium of the knee and watching the drainage through the lymphatics there, but also watching to see the vascularity of the cartilage in the knee as well, and how vascular that is, but also looking at the oxygen saturation as well. So we've had a little bit of experience doing that. It depends kind of what you mean by in detail, but that we have done some of this work.
Dieter, anything you could add there, or is something you've tried, or is Drew's explanation covered more or less your experience?
Drew has covered most of my experience as well, looking at mouse knee rheumatoid arthritis, either induced by injection or just old animals where we could see the cartilage, measure its thickness. But again, I'd like to repeat Drew's question on that. It depends what high detail would mean. For example, cartilage thickness, we were in the, I think, 120 micrometer range for looking at the cartilage right on top of the tibial bone or the femur.
Yeah, and that's something really the high frequency, high resolution ultrasound specifically would give you that sort of ability, because we are able to image down to approximately 30 to 40 microns. So, okay, well that's interesting. With all of these questions, we will be recording them so that if there's more detailed follow-up, we can certainly engage in a more in-depth discussion around some of these topics, which some of them do require quite a bit of detail. Drew, here's one for you, a little more technical. This is kind of a tough question, but if you could speak to it. How does the spectral unmixing account for the changing spectra due to the fluence dependence? So as you go deeper into tissue, obviously the fluence there will be attenuation because of the tissue path. How have we dealt with that? Do we see it actually affecting any of our results with the spectral unmixing, or is it fairly robust to be able to be independent of that attenuation?
Yeah, there are several considerations here. There are changing, the spectra may change because of the change in fluence at depth. It may also change because of the contrast agent that you're using itself. For example, there's some dyes which at different concentrations, or when they have dissolved in different solvents, will actually change their spectra. So I think characterizing your agent carefully in vitro is actually a really nice step to be able to do that first before you go in vivo as far as the contrast agent side is concerned. Now, as far as changes in fluence, which essentially is light energy per unit area, so changes in the energy of the light at different depths, it's possible to apply very general corrections for that, and this has been done by different research groups. We don't do this specifically because it's very easy to do in a phantom. It's very difficult to do in vivo. You've got different amounts of vasculature and different tissues, and it becomes difficult to predict. Just from a really overall, I've done a lot of this imaging, and from an overall, I would consider the changes in the spectra due to the changes in the contrast agent a much more significant factor than the change in the fluence. So what I'm talking about here, we send light into the tissue, it hits the surface of the skin, it scatters, it goes everywhere. So we're hoping to get a more uniform fluence because of that delivery of the light. But also the changes of the agent. You take an agent and you can inject it into the bloodstream, plasma proteins will stick to it, and that could induce different changes. Different agents, if they're not properly quality controlled, can have these changes that occur, and I find those changes a lot more significant than any kind of fluence changes.
I think it's probably important to add too, you showed a workflow through the software that identified the ability to characterize in vitro. There's nothing really preventing you from doing such a characterization in vivo. And if you had a very well-defined assay at a particular depth where you knew there was agent located, you could potentially create characteristic curves to unmix based on that information.
Yeah, that's exactly true. So let's say you haven't, or you're unable to characterize your contrast agent in vitro, and I've done this before, even if I have done it in vitro, put it in vivo, it changes, but then because I can see it, and because I can image across the whole wavelength range, I can actually characterize it in vivo. I can find a little spot where I can see the contrast agent, characterize the curve there, and then use that to do the unmixing. And that's a good point, Andrew, thanks.
Right, well, I'm actually just looking at the time. We're about 3 minutes left in the session. I do want to try to stay on schedule. So we do just have a few announcements to finish things off that I've been reminded of here. The first one is that just in terms of to get more information about some of the news and views that are going on within the multimodal imaging community, and specifically how it relates to what's going on in visual sonics, I encourage you to please check out our Vivo blog. This is a new portion of our website. If you go to our website, visualsonics.com, you'll see a menu item for the Vivo blog. You can view the blogs there, you can subscribe, and I encourage you to please check out that area of the website, which has some new and interesting information that we'll continue to discuss this topic online. The next announcement is to make you aware of our next webinar. You can see the slide here that's up. This is from Dr. Malidi from the Wellman Center for Photomedicine at Harvard Medical School. So here, Dr. Malidi will be talking specifically about cancer research and the clinical translation using ultrasound-guided photoacoustic imaging. So I encourage you, please, to register for this next webinar. Date will be posted soon on the website, but again, if you visit visualsonics.com, we're just putting some of the links up here in the chat window. If you go to our resources section under webinars, you will find more information, and you're free to register for the webinar at that time. That brings us to the end of our session. Again, I'd like to thank both Dieter, who joined us online all the way from Europe. I appreciate Dieter taking the time and sharing some interesting applications and information with us. Drew, also for sharing lots of detail on multimodal and specifically molecular imaging. I thank the audience for all the great questions, and I hope that we can speak with some of you soon. Again, if there's any further follow-up, please do contact us. You can contact us most easily through our website, through our customer support page. And we can certainly follow up with you and discuss more of this later on. Thanks, everyone, and we will be in touch with you soon. Take care.